A very good morning to class 11th. My dear students, today we will start next type of cell division, that is meiosis. Okay, this topic is very important to the point of view of your final exam as well as your NEET exam. नीट एग्ज़ाम में बच्चे यहाँ से मैक्सिमम टाइम्स क्वेश्चन आता है मियोसिस में से. ओके, मियोसिस का जो प्रोफेस 1 हम पढ़ेंगे दैट इज़ वेरी वेरी इम्पोर्टेंट. ओके, सो आई होप यू ऑल विल लिसन एंड अंडरस्टैंड दिस वीडियो very carefully. Okay, so be attentive and just try to understand all the concepts. Try to recapitulate all the diagrams in your memory. Okay, because this topic is very very important for your further higher studies. Okay, so I want that you should clear your concept in this class only, so that you don't face any kind of difficulty in your higher classes. So let's begin with the topic, that is meiosis. Okay, so meiosis is that type of cell division where the number of chromosome it get reduced to half. Okay, it is the type of cell division where the number of chromosomes they get reduced to half. So which is the process that you have already studied in your lower class, where you have studied the process of meiosis. That is during the sexual reproduction, when gametes they are formed. At that time we observe that the meiosis it takes place. Parent cells which was diploid, they give rise to the gamete cell that is haploid. So during this process of sexual reproduction, where the gametes they are formed from the parent, we observe that parents, those were having 46 chromosome, now when they form the gamete they just have only 23 chromosomes. So here we can observe that number of chromosomes they get reduced from 46 to 23, that is completely half. So that's why this kind of meiosis is called as reductional division. Now, so we can relate that gametogenesis, the formation of gamete, whenever it occurs, it will undergo the meiosis or the reductional division. Now, in sexual reproduction, as gametogenesis, it reduces the number of chromosome to half. So these number of chromosomes they get restored again by the help of process called as fertilization. In fertilization, what happens? The two different types of gametes they fuse together. Okay, haploid gametes yani 23 chromosomes, they will again fuse to form 46 chromosomes. Okay, so this number of chromosome it is again regained by the process of fertilization. So this was the mechanism of sexual reproduction, where we can say both meiosis as well as mitosis it takes place. Okay, so now how meiosis is different from mitosis. If we talk about meiosis it is divided into the nuclear as well as the cytoplasmic and further cell division as it was in mitosis, but here is the elaborated process. Now meiosis is not a single cell cycle, it undergoes two cyclic chains, that is meiosis 1 and meiosis 2, one after another. Firstly meiosis 1 takes place, then after that meiosis 2 takes place. So if we talk about meiosis 1, it is started after when the parental chromosomes they have replicated to produce the identical sister chromatid during the S phase. During S phase what happened? The parental chromosome it forms its duplicate, it forms its replica, it forms its identical sister chromatid. Earlier it was like this, and now it has formed its one more pair. Okay, and this is called, these two chromatids they are called as sister chromatids. Now meiosis it involves the pairing of these kind of chromosome. This structure It is called as homologous chromosome. So if we talk about the structure of homologous chromosome, it contains two sister chromatids. It contains two sister chromatids. Clear? So meiosis involves the pairing of two homologous chromosomes and recombination between them. When these kind of homologous chromosomes, two homologous chromosomes, they come near to each other, what happen? Both of these, they get paired, they get recombined. Okay, and that process is called as recombination. So if whenever we talk about meiosis, we will always study about the recombination process. Okay, so at last of meiosis, if we compare it with mitosis, only at the end of the mitosis, two diploid cells were formed. Two diploid cells were formed, but at the end of meiosis, what will happen? Four cells, they are formed, and four of them, they are haploid cells. This is the major difference between mitosis and meiosis. In meiosis, four cells and all of them, they are haploid. In mitosis, two cells and both of them, they are diploid. That is the resultant. Clear? Now if we talk about the different cycles, that it's divided into meiosis 1 and meiosis 2. So meiosis 1 is further divided into prophase 1, metaphase 1, anaphase 1, telophase 1, and likewise meiosis 2, it is divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. Now further, prophase of meiosis 1 is quite different from the prophase of mitosis. How it is different? Prophase 1 meiosis is very much complex and longer as compared to the mitosis prophase. It is much elaborated process. Okay, so prophase 1 of meiosis 1 is further divided into 5 phases. Again, the very important topic for your NEET exam. Okay, you have to learn all these stages in a serial order. Okay, and second type of question is the characteristic feature of each stage, that we will further discuss. These all characteristic features, they are very important for your NEET as well as for your board exam of class 11th. So let's begin with the first stage, that is the lactotein stage. Now this lactotein stage, in this stage what happens? The chromosomes, they start becoming visible under the light microscope. The chromosomes, they start becoming compact. We are able to see the compaction of chromosome throughout the lactotein. So what are the key points of the lactotein? That is, chromosomes become visible and they start compacting themselves. Now the second stage after the lactotein of prophase 1 is zygotine. In zygotine what happens? The chromosomes, they start pairing together, and these pairing of the chromosome, that process of association is called as synapses, and such paired chromosomes, they are called as homologous chromosomes. So this one is one homologous chromosome and this one is another homologous chromosomes. So this synapses, it takes place between the two homologous chromosomes. So synapses of chromosome forms the synaptonemal complex. So when both of these homologous chromosomes, they get synapse, they get paired, they get attached. So this complex structure, it is termed as synaptonemal complex. Okay, so this complex formed by pair of synapsed homologous chromosome. So if we see this diagram, this is the one homologous chromosome, this is the second homologous chromosome. When they get synapsed, when they get associated with each other, they form bivalent, or we can also call it as tetrad. Okay, so at the end of zygotine, we have bivalent or tetrad. The most important feature of zygotine is synapses. formation of synaptonemal complex, okay, homologous chromosomes, formation of bivalent and tetrad. These are the key words, key terms which are related to zygotine. Now these two stages of prophase 1, they are very short lived, they just remain for very short duration. Now we come on to the next stage of prophase 1, that is Pachytine. Pachytine is the next stage where these bivalent chromosomes, they are clearly appeared as the tetrads, and now these tetrads, they will form the recombination nodule. So here in this diagram, if you observe carefully, I have mentioned the sister chromatids, known sister chromatids. If we observe these known sister chromatids which I have shaded, we see that they are associating with each other and forming a point of attachment. This point of attachment and this point of attachment, it is called as recombination nodule. Recombination nodule is the area where crossing over is taking place between the two known sister chromatids of two homologous chromosomes. So what is crossing over? It is exchange of genetic material between the two homologous chromosomes. And what I have discussed with you right now, two homologous chromosomes K, two non-sister chromatids which I have shaded, they will form the recombination nodule, okay, and that process is called as crossing over. Now this crossing over is an enzyme mediated process, and the enzyme which is responsible for crossing over is recombinase enzyme, and always crossing over leads to recombination. It means when these two chromatids, they get crossed with each other, they will exchange their material, they will recombine